Well, now that we know the basics, we'll move on and have Meredith Hullar, who is a staff scientist at the Fred Hutchinson Cancer Center, tell us about her research, which includes the role of the microbiome in diet and human health, and how the gut microbiome metabolizes dietary constituents to alter the exposures that may influence our health outcomes. Dr. Hullar will discuss the epigenetics of the microbiome. There she is.
Thank you so much for inviting me here today to talk to you about the microbiome and epigenetics. So we are not alone. As was previously pointed out, a large proportion of our genomic DNA that's associated with humans is actually provided by bacteria that either are found within the human host or on the human host. And I've been asked today to talk to you about epigenetics and the microbiome. So I've broken it down into two different sections. One is just a brief overview of epigenetics within the bacteria, looking predominantly at DNA methylation. And then I'll move on to looking at some potential epigenetic mechanisms between the host and the microbiome, looking at DNA methylation, non-coding RNAs, and microRNAs specifically. And then I'll talk a little bit about microbial metabolites of diet, which was pointed out as being such an important thing, and how that may influence host health. And then I'll end with talking a little bit about my research in obesity and the microbiome. So as was pointed out early on today, humans have methylated DNA, and it's mostly at the N5 methylcytosine and found in CPG islands. Bacteria have three different types of methylation, methylated nucleotides. The predominant one is the N6 methyladenine, and then they also have N5 and N4 methylcytosine. This is these bases are methylated by DNA methyltransferases, where the methyl group is transferred from the S-adenosyl L-methionine to the nucleotide base. In bacteria, DNA methylation predominantly protects the host bacterium against phage attack. And this is through restriction modification systems, which are comprised of the methyltransferases and restriction endonucleases that cut DNA at the internal phosphodiester bonds, and they recognize unmethylated sequences that are 4 to 6 base pair palindromes. And many of us have used these restriction systems in our molecular biology work. So the way that this happens is that in the cell, methylated DNA here, the host DNA is methylated. When a phage injects its DNA into the host cell, the phage DNA is not methylated, and the restriction endonucleases then can cleave the foreign DNA, and so that it won't be incorporated into the host genome. However, sometimes these phages escape, and the phage DNA is lysogenized into the host cell, and then the phage DNA has the same methylation pattern as the host. And so this phenomena has really led to a diversification of these restriction modification systems. And here you can see that restriction modification systems within bacteria are broadly distributed over many different bacterial phylum, and that there are over 2,000 different systems with 43,000 restriction modification enzymes. When the endonucleases end up being modified, there are new functions that can arise for these restriction endonuclease, or these restriction modification systems. And in this case, one example is DNA mismatch repair, which is an E. coli DNA adenine methylase, the DAM system. And basically what happens is that during replication in the host cell, there's a lag period when the daughter cell is formed. And this mark right here represents a mismatch in the host DNA to the daughter strand that's formed. This mismatch is recognized by mut-S, and then that forms a complex here with mut-L and mut-H, and the endonucleases cleave the nucleotides, then exonucleases remove them, and then polymerase III recreates the daughter strand in a perfect match to the host, to the parent strand. And this is all dictated by the methylated base that is in the CTAG region. There also has been some work that's been done that shows that their methylation changes can also affect gene expression within this DAM system. And it's been shown that, hypothesized that the changes in gene expression are associated with genes involved in phage infectivity, DNA repair, SOS response, culturability, and host association. So these are very dynamic systems. Using some new technology, Krebs et al. Showed that there are a lot of different methylation marks within the H. pylori microbiome, or genome, sorry. And this is just a paper that they did where they were comparing 26695 to another strain, J99R3. And here we have all of the different methylation sites that are happening within the genome. And we have listed here the different palindromes that the restriction endonucleases can recognize. So that's quite amazing that there is this much methylation going on within these systems. And recently, Leonard et al. Looked in 2 strains of Bacteroides dorii, which are the predominant genera that are found in the microbial gut microbiome, and found that there were huge differences between the methylation patterns between the 2 strains. One was very methylated and the other was not. And we know that it's hypothesized that within H. pylori, methylation can reduce natural transformations, but we're not sure really what's going on within the Bacteroides dorii. It could be that differences in methylation, which are leading to differences in gene expression, could be a function of different functional adaptations to different environments. And then moving on to epigenetics that is really between the host and the microbiome. There's quite a bit of work in pathogenesis and that has shown that host response via DNA methylation or non-coding RNAs and microRNAs can change upon exposure either to one infective agent or in response to a complete microbiome. And then I'll also talk about the microbial metabolites of diet. So I think that this is a really interesting example of how gut homeostasis may be regulated. In this system, may be regulated by methylation. In this system, we have the toll-like receptor gene, which is a gene involved in innate immunity. And the gene, the toll-like receptor recognizes gram-negative cell wall material from microorganisms, in this case found in the gut. And what Takahashi found is that there was DNA methylation of the TLR4 genes in intestinal epithelial cells, either in both in vitro and in vivo systems. And they found that there was a difference between the degree of methylation between mice that were germ-free or had no bacteria and those mice that had their full complement of the complex gut microbiome. And what they found is that in the colon, there was gene methylation, which means that the gene was not expressed. And that suggests then that the whole inflammation cascade that happens when a toll-like receptor encounters gram-negative bacteria is turned off in the colon, as a way to establish homeostasis with the natural or the full complement of the gut microbiome. And others have also found this with other toll-like receptors. In addition, upon infection with a single organism, there seem to be changes in microRNA profiles, and microRNAs are associated with expression with changing transcription patterns. And in this overview, it's just showing really that when an organism is infected with Listeria or a Salmonella or Helicobacter or Mycobacterium, there is a change in the host response in their microRNA profiles. And what seems to happen is, for example, here, mRNAs associated with genes involved in the immune response are upregulated. That causes a blockage, and that allows an infection to occur. Similarly, the microbiome can alter the response of the host to an infection. So it matters whether or not you have a full functioning microbiome versus not, versus being infection or germ-free. And in this case, they looked at two groups of mice, one that had a full complement of its gut microbiome and one that was germ-free, and they found that the expression patterns of the microRNAs changed whether or not you had the full complement of bacteria. So this really suggests that there's an interplay between the host and the microbiome, that the complexity of the microbiome really is influencing how the host responds to these different onslaughts. One of the next areas is really a hot topic right now. We're moving beyond just cataloging the bacteria that are present in these different systems and really starting to look at the interactions of gut microbial metabolism on the host. This is the colon, which is the area that I'm focusing on, is really a unique anaerobic environment. It really selects for very, very specific microorganisms which do very different metabolism than the human host does. And the idea then is that they really create an exposure for the human host that we are just starting to take into consideration. And this happens primarily through colonic fermentation of compounds that get down to the colon. So this usually is phytochemicals, compounds that are not taken up in the small intestine. However, in this day of overeating, we're finding that proteins are often making it down to the colon with its whole association of anaerobic fermentation products, which may influence host health. And so here we have carbohydrates that are being metabolized, and they're metabolized down to fermentation products like acetate, butyrate, and propionate. And this happens through a complex interaction, trophic interaction, really, of many different organisms which are in the gut. It's not just one bacteria does one metabolism. Oftentimes, it's a whole consortia of organisms, which we think leads to some of the diversity high diversity that we tend to see in the colon. So then these compounds like acetate, butyrate, and propionate can interact with the human host. And really, if you look in the literature, there are really quite a few compounds that are associated with phytochemicals that undergo microbial metabolism, which then potentially have epigenetic influences on the host. When it's a little more complicated than the fact that bacteria ferment carbohydrates to short-chain fatty acids. In actuality, we saw a lot of diversity in the 16S, but there's also a lot of diversity in the metabolic pathways that bacteria have. So in this case, where we're talking about the fermentation of carbohydrates to short-chain fatty acids, there are 5 different metabolic pathways that can all lead to butyrate, for example, which is a short-chain fatty acid of interest. And the distribution varies from people. Everyone in their gut microbiome, everyone has this pathway, which is really the dominant pathway. They also have the glutarate pathway, but then these other two pathways are sometimes present and sometimes not, so leading to potential inter-individual variation in exposures to the host. So this is an overview of linking the gut microbiome to the host metabolism, and you see, you see here we have microbial metabolism of short-chain fatty acids. These short-chain fatty acids have been associated with satiety, with the peptide YY. The short-chain fatty acids can move into the blood and then undergo systemic circulation and interact in adipogenesis. Acetate is involved in skeletal muscle energy and lipogenesis in the gut, in the liver. One of the other things that can often happen is gram-negative bacteria, often associated with various disease states. You have a relaxation of the tight junctions in the epithelium. These gram-negative cell wall material can be then cross over into the blood and create low levels of systemic inflammation that have been associated with obesity. In a study that by Lukovic et al., this is just an example of looking at gene expression here as influenced by exposure to short-chain fatty acids of either acetate, butyrate, or propionate. And what you see is that there are many genes associated with fatty acid metabolism and with satiety that are influenced by exposure to butyrate or propionate. And then also there's an interaction with histone deacetylase and butyrate and propionate, suggesting that there's an influence here between the gut microbiome and host epigenetics. So in summary, this is a burgeoning area. So the role of methylation is important within the microbiome and also between the microbiome. And also a new area that I think is important to start thinking about is the interaction of diet and epigenetics in the interface between the microbiome and host. I think that we're really hindered at the moment by our sample sizes that we have available. We have no prospective studies, so it's very hard to, I think they're just beginning to be formed, but it's very hard to really say how early life microbiome may influence your obesity later in life at this point. And so just moving on to some of my research, we all know that diet is associated with adiposity through changes of fuel availability and energy imbalance. And this happens through an increase in circulating levels of lipids, adipocytes, macrophages, and inflammation. And there's new evidence that the microbiome is also associated with this pathway. Some of the early work by Turnbaugh et al. Showed that there was an increase in genes associated with carbohydrate metabolism and fatty acid biosynthesis in the metagenome of people who are obese, suggesting that people that are obese have different energy gathering capacity or energy yielding capacity than lean people. So we were interested in whether the microbiome is associated with obesity. And we looked at this in a cross-sectional study of women who were ages 40 to 45. One of the big criterion is they cannot have had any antibiotic use in the last 6 months, or in this case, have any digestive diseases. We were able to obtain fecal samples from 107 people out of the 205 women that we asked. This is a little lower than what we seem to be able to achieve now. And we measured adiposity by DEXA, and we looked at the community structure using 16S ribosomal RNA genes. And then we measured diet. We asked people to complete a 3-day food record, a 3-day food record. And what we found is that we could group women or identify women by their dominant microbes found in their gut. And this is a multivariate analysis, which shows we explained about 30% of the variation in the microbial community with axis 1. But we found 3 different groups, one that was dominated by the genera Prevotella, one dominated by Bacteroides, and one dominated by Pyramidobacter. And this is something that's been found in other studies, although this grouping is a little different. And it turns out that perhaps some of the functional capacity of this group is similar to some of the other groups that we've found. So we also found that when we looked at the distribution of, or the associations with diet and phylum of bacteria, we found that Bacteroides was associated with increased animal protein intake and decreased fiber and vegetable protein intake. And we found that firmicutes were associated with increased fiber intake and increased vegetable protein, which is similar to some other work that's been done by Gary Wu's group. And then when we divided the women into tertiles of adiposity, we found that there was a significant difference in an inverse relationship between the amount of dietary fiber the women took in and an increasing adiposity. And then when we just modeled this association, we found that even after we corrected for fiber intake, there was still a significant association between the microbiome and adiposity. So moving on, I'm currently involved in a study involving the multi-ethnic cohort, and this is the overarching theme is to look at obesity, fat distribution, and disease risk. And this is primarily because health risks due to obesity have been associated with the different types and location of adipose tissue. And there are also strong ethnic and racial differences in BMI and cancer associations that are linked to these different distributions of adiposity. And in this grant, we are actually measuring adiposity using MRI and DEXA. And then we have, this is a large program project with Loic Lemarchand. There are 4 different groups here, and we're going to look at the exposome in Project 1, look at genetic variation, or our MWAS, as we're calling it. Also look at metabolomics and look at the microbiome, and integrate these different measures to look at predictors of adiposity as associated with disease risk. And we're just starting this. At the end, we should have about 7,000 participants. We're about 3,000 into it at this point. So we're very interested and excited to see how these data turn out. So a lot of this work has been done in conjunction with Johanna Lampe at Fred Hutch, and then these are the other people that have been involved in the research. Thank you.